on, just to briefly introduce Eric. Eric is a recent assistant professor and pretty much confident he will soon be promoted to a position at the Institute for Center for Environmental Science and Technology, Maryland University of Maryland. I first met Eric back in the early 2000s at the time when he was working out on crabs. He used to work on oyster persistent parasites. And at a conference where the entire day of talks was on the prevalence and abundance of some kind of parasite oysters in different places around the nation, Eric struck me by the only dog who's been looking at mechanism explaining the disease processes. That was pretty unique at that time in this area, in this research. So it was pretty clear to me that this guy, and once I learned more about Eric, where he comes, what is his background, that can be clear that he's bringing really new perspective to this field of research. Eric got his PhD degree in medical sciences from Harvard, working on pseudomonas. Then moved to postdocs, couple of postdocs, one on plant science in New Zealand for a little while, and then the other on yeast genetics, before started working on oyster parasite, and then moved on to other parasites, marine organisms. Today we're talking about some new discovery on an old pathogen, because the pathogen is known for some time, although Eric brought his energy, perspective, and unique thinking, I think, to really discover new, new horizon on the importance of this biology. Eric,
thank you, Bassem.
Thank you all for being here, and thank you for the invitation. That was, I mean, it's really a pleasure to be here. So there are a lot of visiting students. I'm a visitor, and I'm just pleased as can be to be here. I think this is a fantastic institution. So there's my advertisement for Stony Brook. I have one more advertisement, and that's for my institution. And that is this summer, we're going to be hosting the International Marine Biotechnology Conference in Baltimore. And I'm just going to flash this up here, and you can either look it up at the imba.org if you want to jot that down, or you can talk to me or ask Bassem later about going to this conference. But I hope that we'll get somebody from Stony Brook down to Baltimore. I'd love to host anybody there. So I'm going to go through a lot today. I'll apologize once, and then I'm just going to do it anyway. I'm going to try to, this is where I want to get you, and that is that the rediscovery of this pathogenic virus has implications in a number of areas, from fisheries management to the understanding of taxonomy of this group of reoviruses, to really all the way to marine population connectivity. And this is, as Bassem described to you, the early stages in my evolution from someone who studies medical science to someone who studies environmental science. And it's almost complete because I'm now collaborating with oceanographers. But the overall message is that I think that the disease has the potential to affect really marine animal abundance everywhere. And now we're oh, what did I do? Oh, we're doing a Java update. I don't think we want to do that. So I'll just talk. And I think that this is one, this system, this virus system, is one system where maybe we have some of the tools to start going there. So I'm giving you a little background on blue crab ecology, and then launch into the discovery or rediscovery of this pathogen, looking at its prevalence, looking at it where we found it first, which was in soft-shell crab production systems, seeing the fishery implications there, and then I want to get into sequence analysis. I'm at a biotechnology center. I've got to tell you some genome stuff, and then how that genome fits into the biogeographic studies. So first of all, just to introduce the blue crab, it's in Maryland we like to think of it as our crab. It's our state crustacean, but we share the state crustacean with 2 hemispheres in about 30 countries. So it exists from maritime Canada all the way to Argentina. And it supports very valuable fisheries in the United States, but not just here, but also in South America, Venezuela and Brazil in particular. I guess, and I'll point out, it has a big role in the benthic ecology in those places as well. The life history of the blue crab, I'm going to put it in my context, which is the Chesapeake Bay. So here I'm up here in Baltimore. A crab that's in Baltimore, if I find a crab in Baltimore, and we find many, they actually started their life out in the ocean as a zoea stage. Okay, those zoea came from mature females who were holding eggs for about 2 weeks until they matured, and those eggs hatched and those larvae hatched out here and were released into the coastal zone. These guys are not osmoregulatory, so they have to live in the coastal zone. And after about 4 weeks, they can find their way back into coastal bays or major estuaries as they metamorphose into this megalopus stage. It's like a little lobster about 2 millimeters long. And so there's a salinity barrier for this part of the life history. Once they make the first crab stage, about 3 millimeters size, they grow and disperse into these estuaries, and they can live in fresh water once they've reached this stage. They can osmoregulate. The role in the food web in our area is really crucial. They take a lot of the primary consumers or secondary consumers from the benthos, and that's, they convey those to a lot of fish species. It's prey for many, many fish species. So it's a benthic pelagic link. I put this circular arrow here because crabs also are highly cannibalistic, and that may be an important part of their life history, that a lot of crabs actually eat other crabs. If you think about harvesting carbon from the environment, crabs that eat other crabs are actually sort of widening their net through their brothers and sisters, I guess. But the biggest predator of blue crab in the Chesapeake Bay are humans. Of the mature crab population, I think about half of the crabs are fished out every year. So that fishery in Chesapeake Bay, the average, it goes up and down, as I'll show you, but the average commercial harvest is about 62 million pounds. That's in Maryland, Virginia, and the District of Columbia, valued around $64 million a year. Okay. It's practiced by, between the 3 districts, about 5,000 fishermen have commercial licenses, and those licenses represent at least 500,000 traps. So you can see the fishing effort in our area is pretty intense. So how do you manage that? Well, the management for the fishery in our area is, and by the way, blue crab fishery management in the United States is not a national issue. It's a state-by-state decision. In the Chesapeake Bay, there's sort of a coordinated, let me move this up. There's a coordinated management between Maryland and Virginia. And there's, let me just go through this. There's a size restriction, so there's 2 things that do that. There are coal rings that let very small crabs escape from the traps. And then there's like a 5-inch size limit carapace width for crabs in both states. It varies from year to year and from state to state, but it's 5 to 5.5 inches. And then we also don't allow harvest of egg-bearing females in either state. And in both Maryland and Virginia, there are parts of the year where you can't take females at all because they're headed to the spawning grounds. I want to use this opportunity to point out something else in this trap, and that is crab-to-crab interactions, which I'll talk about later as we talk about disease transmission. So here's a crab grabbing the leg off of another one. And probably as these crabs come out of the trap, that leg is going to still be in this claw, although it won't be attached to its crab anymore. So here's this variation I mentioned in crab harvests from 1990 to 2014. And I'll just point out the last few years. We had a crab harvest of over 80 million pounds in the Chesapeake Bay in, 2010. And in 2014, it was about 35 million pounds. So it varies quite a lot year to year. The states try to target mortality so that you're getting more males than females. But if you see overall, as I mentioned, if you average this out, 33% plus 25%, if you have an even sex ratio, you're well over 50% fishing mortality every year. So what is this? Why does it vary so much? Well, not surprisingly, it varies a lot because the crab abundance itself varies a lot year to year. This is a chart from the Chesapeake Bay Stock Assessment Committee, and they collect data from what's called the Winter Dredge Survey. And the Winter Dredge Survey is one of the most intensive fishery independent assessments of abundance that I know of. It takes place throughout the Chesapeake Bay all winter, and 1,500 dredges are done throughout the sediments to see where crabs are overwintering. And they take everywhere from sort of thumbnail-sized crabs up to the adults. And all this trawl data and dredge data gets plugged into a big model that takes into account a lot of the life history factors and catchability factors and comes up with an overall abundance. And that's what management decisions are based on. So, this is the foundation of the harvest variation, but now it just begs the question, which is like, okay, well, what's the foundation for the abundance variation? And I'm not really going to answer that question for you today. I'm just going to talk about how we sort of slice that up. Well, I mentioned sort of on the reproduction side, we mentioned those restrictions on collecting egg-bearing females or females at all. So you're trying to preserve your spawning stock. And then changes in fecundity, that is how many eggs or how many larvae are produced per female, is also going to affect your reproduction rate. Blue crabs can actually set multiple sponges. They mate once in their life, but they can set multiple sets of eggs. So each time maybe 2 to 3 million eggs, and they can do that up to 5 or 6 times. So depending on whether they get to do it once or twice or 3 times can change their fecundity. On the mortality side, there's lots that we can think about. Starvation and environmental stresses on those larval stages could be pretty intense. Predation and fishing are biggies. I've done a little work on toxins, but today I really just want to focus on disease. And that disease, I'm going to represent it in another way. If you look at the life cycle of the crab, we're going to be looking at this region of the life history between the juvenile and mature crab stage. But overall, the life history, you may have several million eggs released, and really you only have 4 or 5 crabs coming out of that. So you have like a 99.9999% mortality rate. My interest in diseases and my belief that diseases probably drive a lot of variation was recognized by the Chesapeake Bay Stock Assessment Committee in 2013. In their reports and in the subsequent reports, they said in sort of their data gaps section, the critical data needs are to analyze the magnitude of other sources of incidental mortality, specifically sponge crab discards, disease, and predation. So I think that helps me writing papers and helps me writing grant proposals to get funded for the kind of research that I'll tell you about. So diseases, or diseases in general, and the pathogens that cause the diseases. There have been some fantastic experts in the field, from Robin Overstreet to Phyllis Johnson to Jeff Shields, other people who've really chronicled a lot of the dozens, if not hundreds, of different diseases that affect blue crabs. And so where do you start? How do you decide which disease is going to be the most important? I've worked on a number of these, mostly Hematodynium perezi and viruses. And today I'm really just going to tell you about viruses. But when I talk about disease at the end, think about this virus as being one of several diseases that might be impacting blue crab populations. So I'll start it out as a story. The story starts here, actually in Baltimore, at IMET, right here on the Inner Harbor. We had a research crab hatchery that was designed to look at the potential for doing restoration breeding, putting juveniles out into the bay. And in our area, as in a lot of the southeast, people produce soft crabs, and I'll explain what that is. But this is a local soft crab producer on a creek in Baltimore. And these are both settings where blue crabs die a lot. So if I'm looking at mortality, I'm going to go to where the mortality is happening rather than out in the wild. Our hatchery was based on taking wild broodstock, that is mated females. They weren't egg-bearing, but they were mated females. We put them in the right conditions. They would extrude a mass of eggs, and then when those eggs matured into larvae, we'd grow them up into the megalopa, and then those megalopa would be grown into juvenile crabs and used for research and ecological studies. We had a lot of mortality in our broodstock. I write here over 50%, but I'd say it's something more like 90% of the broodstock that we brought in and put in quarantine and then into our hatchery system ended up dying within a couple of months of arrival. And it was a real mystery as to what was going on. And if we look at soft crabs, the soft crab system, this is the practice of taking small pre-molt animals, waiting until they pop out of their shell, taking that soft animal and then refrigerating it and selling it as a value-added product. It's hard to give a sense of the scale in Maryland because the state doesn't keep any records on how many people are doing it. But if you look at National Marine Fisheries data and some state data, maybe 2% of the national harvest goes to soft crabs, but it represents possibly 10%. And in the Bay, talking to our managers, they're guessing that 5% of the harvest goes to soft crab production. Well, a lot of crabs die in soft crab productions. Typically, you get more than 25% of the animals die. So they'll take pre-molt animals, put them in these shallow tanks with a flow-through system, pulling water out of a local creek, or maybe doing a recirculating system through a filter. And they talk about success. So they'll have 75% success or 70% success, but that means you have 25% or 30% mortality. And you can look at, it's not hard to imagine the reasons why this is happening. They're high density, they're about to molt, they're possibly extreme temperatures. And of course, I'm going to tell you that they're actually all infected with a virus. But I just want to point out, I've tried to do a back-of-the-envelope calculation of how many animals die in these systems in the Chesapeake Bay alone. And I'm guessing that if it's 5% of the harvest, if you go back to the chart that I showed you what the average harvest is, it's between 1 and 2 million dead animals in these systems alone every year. And some of these systems can be quite large. This is a Google Earth shot of one in southern Maryland. This is a dumpster here. So these are facilities. This facility here has double-decker shedding tanks in it, probably a couple hundred, if not more, tanks here. There's another one here, another one here. So there are probably 1,000 shedding tanks in the system, and they're all flow-through. I don't know if I have a picture of that. I don't. But they're all flow-through. That is, they're pulling water out of the estuary here, and they're just pumping it right back in after it goes past the crabs. This facility has dumpsters for their dead loss, which is unusual for the industry. That most people throw them back in the water. So I looked at these situations where there was mortality and said, well, what's killing them? Well, we could rule out a lot of other causes, injuries and stresses and water quality. But when we take those away, then you need to think pathogens. And you can look histologically and see that there were no metazoan or protozoan parasites in most of these animals. So if it's cryptic, I'm assuming it's going to be a virus. And what we learned from shrimp aquaculture is that mass mortalities in these intensive settings are often caused by viruses, and they arise sporadically, and they're intense, and they cause quantitative death. Well, the literature had several viruses described in blue crabs, and the only ones that were seen to be pathogenic had RNA genomes. So, and this is where I'm getting into the biotech side, the molecular side. And if you look at the genomes of the 3 types of viruses that have been described in blue crabs, there had been a reovirus described, a rhabdo-like virus described, and a picorna-like virus described. This is all based on electron microscopy from the 70s and 80s. And that was really the state of the art. That's how you described the viruses at the time. It was the best that you could do. And in fact, the first virus described in a crustacean ever was, I think, in 1966, when electron microscopy got to the point that someone could grind up a sick crab and put something on a grid and say, look, there's a virus. So this was the new technology at the time. So these had all been described in blue crab as possibly being associated with pathologies. But if you're familiar with eukaryotic biology or bacterial genomics, those systems have genetic handles or shortcuts that you can take to identify genes or identify a species just by using conserved oligonucleotide primers to amplify a gene of interest. Viruses don't have that opportunity, don't provide you that opportunity because their genomes are too diverse. There's no homology across viruses that you can use to hunt in that kind of a way. So we had to go back up even further and say, is there something, though, that's common about all these virus genomes that we can use as a handle to grab it out? And that commonality was that they have an RNA genome, okay? But they're RNA genomes that are plus-stranded or minus-stranded or double-stranded. But they do have one commonality, and that is in their replication cycle, they all have to go through a double-stranded intermediate. And that is, and I'll just explain here. If you have a double-stranded RNA genome, like that reovirus, just like our genome, that genome is going to go through semi-conservative replication. That is, the positive strand is going to be the template to produce a negative strand. So this blue strand here is going to make the negative red strand during genome replication, and vice versa. So double-stranded RNA genomes are quite easy to understand. But if I want to make a new single-stranded RNA, positive single-stranded RNA genome, I have to make that negative strand, and then that's the template to make a new positive strand, and vice versa for a negative strand. So no matter what sort of genome you have to go through at least a little bit of double-stranded intermediate. So that's what we use. That's what we use to try to get a handle on what virus might be present in these systems. And it comes down to the fact that chemically, physicochemically, the double-stranded RNA is very different from both DNA and different from RNA. And it was actually quite easy to pull double-stranded RNA out of nucleic acid extracts from crabs. So that's what we did. Working with that crabber, whose crab shack I showed you in Dundalk, Maryland, They identified for me crabs that had died in their soft shell facility, and we took healthy ones and dying ones, ruled out other causes of mortality, and investigated what the nucleic acids looked like in those crabs. And the very first crab we pulled out that met those criteria was what I'm going to talk about for the rest of the talk. And that is, in a dying crab, we extracted total RNA and then enriched the double-stranded RNA from that. The D is for the dermal layer, that's the dermis just below the shell. H is for hepatopancreas, the digestive organ. And M is for muscle. And in all three of these tissues, what we see was a laddering of double-stranded RNA in the dying crab. And in the healthy crab, we saw no such laddering. So this was our first indication that we may have a virus and that it may be associated with mortality. But that was just an N of 1. It also told us from the banding that this looked like a reovirus. So this gave us now an assay, right? We can now use this double-stranded RNA assay to investigate mortalities from shedding facilities anywhere that we want. Oh, I'm sorry. And we did investigate this guy and showed that we had virus-like particles in the tissues that we had extracted. So we prepped some of it for histology and some of it for molecular biology. This is kind of an ugly picture because it was actually prepped for histology, and then we kind of used the histological prep for an electron microscopy prep assay, which is not an ideal fixation condition. But if we now apply this, just the double-stranded assay, not microscopy, and look at mortalities from soft shell systems from the Delaware Bay all the way down to the Gulf Coast, and this is our assay, right? Just do we have bands on a gel? We found that 50 to 90% of the mortalities that came out of every soft shell system we saw had this banding pattern. So this was actually a surprise, but it really did confirm that we have an association. So you can imagine that crabs that are dying may just have the virus because they're dying of other reasons, and the virus has taken an opportunistic route in, and it's not the cause of death. We also looked at our hatchery. Remember I talked about all those mortalities there. We had been saving a few of the unexplained mortalities in the freezer, and we took them out, extracted RNA, and looked at double-stranded RNA in those guys as well, and found that both the blue diamonds are wild broodstock that had died, and the stars indicate hatchery production. These are juvenile crabs that came out of our hatchery that had a massive unexplained mortality as well. And so I think what this gel indicates is that a lot of our broodstock had the virus, and every crab that was involved in that massive mortality, every juvenile crab had the virus. So this indicates that it was coming in in broodstock and got into our hatchery production, at least in that one case where we saw mortality. So as I described, this is a redescription. What we really found or rediscovered was a virus that Phyllis Johnson had written up in 1977 as Rio-like virus, which is a rather nonspecific name. So we renamed it CSRV1, and I'll explain why it's CSRV1. So the question for us was, why was this not recognized earlier as being important in mortality? It had been described in the literature, but people didn't understand how widespread it was. And it really comes down to not that many people were looking. It's hard for me to believe that not that many people care about why blue crabs die. But really the reason was it was histologically cryptic. I've worked with the person who has Phyllis Johnson's position at the NOAA Oxford lab now, Gretchen Mesick, who sent me histological samples that she says she can't see any virus in, and I can pick it up. I'll show you later. I'll just tell you. We can pick up by PCR that there are maybe 10 to the 8th or 10 to the 9th viruses per milligram tissue in samples that are histologically negative by its typical H&E staining. So I think we just had a virus that was hiding under the radar. The reason that there's a CSRV1 is that using the same assay, the same double-stranded RNA assay to look for virus, to do virus discovery, we have found another reovirus, which we're going to call CSRV2. But we found a whole bunch of viruses in crabs throughout the Atlantic coast that we can't identify. They don't have banding patterns that look like anything that's in the literature, and they don't have genomes that look like anything that's in the literature. This crab here, AR32, stands for Agawam River 32. We found this in 30% of the crabs from one river in the South Shore of Massachusetts one year. We've never seen it again. We've sequenced the genome, and the genome doesn't look like anything in the GenBank database. So this method is good for finding new viruses, but we need to start to connect the dots on these. Anyway, today I'm just going to talk about CSRV1. All right, so I told you about the association between CSRV1 and mortality, but we really need to satisfy Koch's postulates and say, does the virus cause mortality? And when you infect a crab, do you get more virus back? And that's what this experiment was designed to do. And this is one in which we injected a filtrate from an infected crab, homogenized an infected crab, filtered that to virus-sized particles, injected that into naive hatchery-produced crabs, and I wouldn't be showing you this if it didn't work out this way, that within 3 days of injecting it, you can see virus banding pattern in the tissues of the recipient animals. And then over the next couple of weeks, it just intensifies, and actually in our mortality curve on this experiment, pretty much all of our animals in the experiment were dead by day 18. So it was quantitative mortality, and all of them showed the virus. We've used both the double-stranded assay and a PCR assay, which I'll explain in a second, to also show that you can transmit this virus between crabs by cannibalism, and that they can acquire it by being housed in one of those soft-shell flow-through facilities for several days. So they can pick it up through cannibalism as well as cohabitation in an environment where the virus is present. When we think about transmission of a virus, we also want to think about, is there vertical transmission? And we don't have a really solid answer on that, but we want to know whether transmission happens from mother to larvae. And I don't have a lot of information on that, except that we do have anecdotal information that when we take egg-bearing females out of the wild, they rarely survive long enough for their eggs to mature and hatch and produce larvae. So we think that that may be a barrier to vertical transmission by itself. But in addition to that, what we've seen is that in one instance, a female that was infected did produce larvae, thousands of larvae, but when we test those larvae, they were negative for the virus. So I think that my hypothesis at this point is that we don't have vertical transmission, and that's an important feature for what I'll discuss later. Our quantitative PCR assay, you're familiar with gene expression or pathogen assays. You've probably seen figures like this. We're moving now from our double-stranded RNA assay to something that's much more sensitive. To develop this quantitative PCR assay, we cloned one of the smaller segments of the genome, sequenced it, and found that it matched nothing in GenBank, and then developed a PCR assay for that segment. And then in a quantitative situation, we can detect from 1 copy to 10 million copies very reliably. So we have a really nice assay for picking up the virus in RNA from whatever you want to extract. And the question for us, after seeing all that mortality in soft-shell systems, was, is the virus out in the wild at the prevalence that we see it in captivity? And thanks to a really big multi-partner group of people, from Massachusetts Division of Marine Fisheries to estuarine reserve people to folks at University of Del State, we got samples for 2 years along the Atlantic coast from these 5 sites, as well as the NOAA Oxford lab provided us samples from their integrated assessment of the Chesapeake Bay from sites within the Chesapeake Bay. And the work of a lot of interns and really hardworking and gifted high school undergraduate and graduate students, what we found is that the prevalence of the virus along the coast and within the Chesapeake Bay is incredibly variable. It goes from 0 to 78%. But I guess the take-home message is that of the 15 samples we took, 2 years along the coast and 1 year in the bay, only 2 of those times did we fail to see virus in any of the samples. The sample size was anywhere from 25 to 75 crabs. And the prevalence could get quite high. If you look at this location here in New Jersey, in 1 year we had prevalence of 3%, but the following year we had a prevalence of 78%. And this was a pretty shocking prevalence rate, and believe me, we checked that a couple of times before we published it. But the overall, if you take all that variability in time and space, and you take the grand average, both inside and outside the Chesapeake Bay, what we see is that outside Chesapeake Bay, all these sites 1 through 5 average to about 22% prevalence, and inside Chesapeake Bay, 20% prevalence. It was an equal opportunity presence in all salinities, both sexes, no correlation with latitude or crab size. So we think that it infects pretty much across the board. And we found, because it was a quantitative assay, that we were getting viral loads of up to 10 to the 8th viruses per milligram of host tissue. So that would be a milligram of muscle tissue from a heavily infected crab has 100 million viruses in it. And these were crabs that were caught very active. They weren't moribund. They were pretty healthy looking. Remember I mentioned the broodstock and the screening that we have to do for our broodstock now in the hatchery. So we found that a lot of them were infected. That meant that now what we need to do is when we bring broodstock in, we pull hemolymph from them. You can take a syringe, pull hemolymph from the animal non-lethally, make RNA from that, and test it for the virus. And what we found there from 2013 and 2014, these are our broodstock collections, is that the fraction of animals that are infected in broodstock was actually higher than the general population of animals that we were sampling in that study I just mentioned. So I'm not sure. There's something going on with either animals that are that mature or possibly animals that are in a reproductive state. But what we find is that the average, the mean prevalence of virus in broodstock and spawning stock animals is almost 50%. And you recall, going back to our hatchery production, this really starts to explain why we had so many animals dying in that hatchery, because they are coming in with the virus at a high prevalence. We're also interested in this study in understanding whether crabs that are virus positive, how long do they live? How long does a crab that's infected live? And so we took this opportunity, since we were making the effort to bring broodstock in, of taking some of the animals that were infected, the ones that you're seeing on these bars here, and instead of putting them in the freezer immediately, we put them in individual tanks separated from one another and maintained them until they died. And what you see from that relationship, is this graph here. We have a graph that shows on the y-axis how many days it took them to die after we got the PCR results, which was a couple days after they came in. And then the log 10 virus genome copy number on the x-axis. So it goes from 1 copy to 10 to the 9th copies. And it's a pretty big scatter, but you can put a regression line through it that explains whatever. It's 0.4 r squared on that. So that's explaining a lot of that variability there. And I think my look at this suggests that we may have something like a threshold effect, that if you have above 100,000 copies of the virus, you don't live very long. If you look here, above 10 to the 5, you probably live 20 days or fewer. And below 10 to the 5, you may live as long as 74 days. So I think there's probably a threshold, a pre-patent period, and beyond that, they just progress to death. And in this experiment, the mean time to death was 24 days. So I just want to take that prevalence, with this 20% prevalence, and our mean time to death, and if we think about that, it would suggest that if this is happening in the wild, that you have a lot of crabs dying in the wild every year from one virus. I'm not going to pretend that this is actually happening. I think that what happens is that you bring these animals into aquaculture, and there's certain stresses that weaken them and make them more susceptible to mortality from this. But if you just look at the numbers as I've described them, 400 million crabs in the bay would be a typical crab abundance. 20% mortality every 4 weeks over a 5-month period, say, from May through whenever the season ends. The math tells you that you could lose 2/3 of the crabs to one virus. I'm sure that's not happening, but we don't know. Here's my pitch to anybody that might be reviewing my grants, and that is, we have a proposal out to NOAA right now to do the only experiment I can think of that would answer this question, and that is a tag return study. You compare tag return rates for virus-infected crabs versus virus-free crabs, and I'll explain that in a second. And we can also take them and put them in an outdoor cage setting that's a little closer to a natural environment than our aquaculture center, and see how long an infected crab lives. But I just want to explain this first part here a little more. It would work like this. Crabs can be tagged easily with a plastic tag with a reward on them. When fishermen catch that tag and they see the reward on it, they're like, okay, I'll get the $5 back or the $10 back. I'll call DNR or the scientist and tell you where I caught it and when and where I caught it. That's all we want to know. And then you send them the money. But if at the time that you put the tag on, you take a sample of blood from the leg sinus here and test that for the virus, then you have a virus titer that you know on that crab. And then later you can compare whether the infected set gave you the same return rate as the non-infected set. So I had to think this through, and I basically drew up some mock data that would say, what would that look like? So if you had a typical tag return study, you'd have in week 1, you might get a certain number of crabs back. You release 3,000 crabs into the bay. Each week you get some back from fishermen, and it drops over time because they're fished out or they disperse or whatever. So your data might look like this from the non-infected set. But if you look at the infected set, our prediction is that the returns are going to be fewer for the infected set, and it's going to decrease. The decay is going to be faster, and it may be if all the crabs die, if every infected crab died in 4 weeks, then in fact, by week 5, this green bar would just not even exist. You'd have no returns. Okay. So we don't know if that's going to be the case. We're waiting to see if we get the funding to do it, and fingers are still crossed on that. But my partners at the Smithsonian Institution, who do the fisheries part of this, the trawling and a lot of the data analysis, put together, sort of taking this idea, they put together what they think would be a predicted tag return rate based on tens of thousands of crabs that they've tagged and had returned over the years. And then they said, well, what would happen if you imposed a certain amount of mortality on that? So their prediction would be that a linear tag return decay would look like this. So if you look at the first bar in each cluster here, we have weeks after tagging versus weekly return rate. And this is based on their real data. What they would predict is in week 1, you'd get a certain percent back, and then it would drop in a linear fashion over time. What they actually see is the last bar in the cluster, that it goes from this number, and it drops much faster than their modeled prediction. And it isn't clear why it drops, but what they've done here in this figure is said, well, we're going to put in a certain mortality rate, a sort of, they're calling it V, I suppose, for virus, but it could be any cause of mortality, that what's really happening is they're seeing that your predicted rate isn't observed because you fish a certain number, but then a certain number of crabs just die for whatever reason. And then you never get that back. So in our case, we're going to peel this apart and say, well, we're going to understand whether that reason is the virus or some other cause. So fingers crossed on that. Remember I mentioned the flow-through shedding systems and that effluent. This is a project that graduate student Emily Flowers just completed with Sea Grant funding, and we were looking here at whether these effluents are making a difference in the prevalence of virus out in the wild. We had this result that a crab that was put in the shedding system can acquire the virus from that water. Now the concern is, well, the crab that's outside and just receiving that, is it going to be more, is it going to have a prior prevalence? So it was a simple study to ask whether effluents, and in this case discards, a lot of the crabs that get thrown out, that get taken out of here, just get thrown in the creeks. Does that influence prevalence? And the study was fairly straightforward. We had 2 sites with commercial scale soft crab production in Deal Island and Crisfield, Maryland. And we wanted to look at these sites as well as a reference site, which is at the Road River, where the Smithsonian has their Environmental Research Center. And in 2013 and 2012, we trawled 2 times or 4 times each summer at these locations. And we looked at each location right next to the shedding facility. So the yellow X's mark where that large-scale shedding facility I showed you with the Google picture, that's at one of these sites. And then we went a couple miles away to a pristine watershed. This is a wildlife management area. This is a state park. So there's no shedding going on here, and it's a much cleaner or much less impacted environment. And the simple question was, is virus prevalence higher closer to the shedding facilities? The short answer is yes. But because of the variability that we got in our sampling, that even though the averages look pretty striking, the standard deviations, which I don't show on here, were large enough that we don't have, just as an aggregate data across 2013 or 2012, we don't have statistical significance. What Emily did show using more sophisticated statistics is that anytime there was an outbreak, that is, anytime you saw prevalence that was higher than the grand average of the whole experiment, it was always closer to the shedding facilities. So there is a statistical significance association with the shedding facilities. It's just not as straightforward as I'm drawing it here. So this seems pretty clear. What about the Road River? Well, the Road River, it kind of messes up the story for us, which is the Road River, where there is no shedding going on, but there's a lot of fishing, but we don't have any shedding facilities. In both the samplings in 2012 had greater than 30% prevalence. So it's not like 30% is an average of two very different numbers. 30% is an average of two very similar numbers in this case. And then in 2013, we had 0. So so it couldn't be more different here. I think what this tells us is that, yes, the shedding facilities have an influence, but certainly, I think as we learned from that Atlantic coast study, you can have high prevalence for lots of reasons. I want to point out one observation here, and that is this great difference in prevalence between 2012 and 2013. If you look at the Road River, it couldn't be more different. And even if you look at 2012 and 2013 at the shedding facilities, there's a very big difference in prevalence. I failed to point it out to you, but in 2012, maybe I did, there were about 800 million crabs in the bay. And in 2013, there were about 300 million crabs in the bay. So for me, it's tempting to draw a connection between the abundance of crabs or the density of crabs and the prevalence of a disease that kills them. We don't know whether the relationship would be that the disease is causing a change in the abundance, or whether that high abundance is actually causing a change in the prevalence of a virus, because you can think of density-dependent transmission as being operative there. But just to summarize the shedding study, again, I didn't show it to you, but extreme temporal and spatial variability. The variability I explained on the coastal range over the tens to hundreds of kilometers in like months to years scale seems to be mimicked even at the 2 to 3 kilometer and 2 week scale, that we would see changes in prevalence over a 2 kilometer range that was 30% from one place to another. And over 2 weeks, that could change by 30%. So this is a virus that comes and goes very rapidly. But tantalizingly, the mean prevalence for 2012 was again that 20%. And in 2013, that low abundance year for crabs was down to 6%. And the only other thing to add here is that we did confirm proximity to shedding. Larger animals had a slightly higher prevalence, statistically significantly higher prevalence. And Emily also observed anecdotally that a lot of the crabs, she was working with a lot of small-scale artisanal soft crab producers, and pretty much to a man, all of them took their dead loss, put it in a bushel basket, and when they went fishing on the fishing grounds, those goes back in the water. If you're a fisherman and you're taking a resource from the environment and you're not thinking about pathogens, it just makes sense that that's where it goes, right? You're putting the resource back where you got it. It would be the responsible thing to do, actually. But the results of that Sea Grant study and these observations that Emily made really led to this proposal, which we're working on now. This is our current main project. And it's being done in collaboration with VIMS and Louisiana State. What we're looking at is, remember I mentioned these interactions in traps, is whether or not crabs interacting in traps are passing the virus from one to another. And also, what is the risk of those discards in terms of passing virus out to crabs in the environment? So just this week, we got crabs I brought actually crabs up from Florida, and we're going to start doing some of these trap experiments. And you can imagine they're simple in design. You'll take a known number of infected crabs and put them in a trap with a known number of non-infected crabs. Everybody's labeled. You let them interact for X amount of time, and you take them out and see whether everybody picked up the virus. So I'd love to talk to you all again sometime and tell you what the result of that is. So we're going to, just to recap that, we're going to measure the potential for CSRV to be transmitted from dead peelers into wild crabs, and different harvest practices to promote that. And that is, there are various ways to harvest crabs. Trapping is the most common, but in the Chesapeake Bay, maybe 10% of the crabs are also harvested on what's called a trot line. And that's a baited rope that has pieces of fish or chicken on it, and you just lay out like a kilometer of that, and crabs will grab onto the bait, and they hold onto it so greedily that as a fisherman, when you pull that line up onto your boat, they're hanging on, and they're ready that you just drop them in your boat. But you can imagine the disease transmission potential for crabs that are independently hanging onto a piece of bait, several meters apart, is much less than those that are in a crab having agonistic interactions, fighting over the piece of menhaden that's in there. And another important piece here, especially I think for our funders, is that it's important to work with fishermen and understand what they would like to do with the information that we come up with. Scientists often are seen to have an antagonistic relationship with fishermen because a lot of times our scientific information gets used by managers to do things that fishermen don't want. And in this study, what I'd like to do is work directly with fishermen, have workshops with them, and say, well, here's what we've learned. You guys can decide what you'd like to do with it. I want to sort of, change that relationship so that scientists don't get grouped with the regulators quite so much. I don't know what it's like in New York, but in Maryland, we're viewed with suspicion. Okay, finally, last few minutes, I want to talk about genome sequence analysis. The short version is that the USDA got a new sequencer at their Iowa facility and heard about this virus and said, we'd like to sequence your viruses on our new machine. I said, that's great. And then Emily, my grad student, said she'd like to assemble the genome, so she did that. And the genome is going to be, we have sequenced the genome. We don't have a lot to say about it. It's going to facilitate the kinds of cellular and biochemical studies that our center is good at, really digging into mechanisms, understanding what the proteins encoded in that virus are. I will tell you about how this changes our understanding of the reoviridae, and I'll get into some of the biogeographic implications. So this is the taxonomy of non-turreted reoviruses right now. So some reoviruses have little projections that stick out on the electron micrographs, and those are the turreted ones. This is the non-turreted ones. You've all been infected by rotaviruses as children. Diarrhea, sniffling nose, cough, whatever. And we all have acquired immunity to that. But those rotaviruses infect mammals, and orbiviruses also infect mammals. These are often livestock pathogens. There's one called blue tongue virus. It's quite famous it causes a disease in livestock. It's vectored by a biting fly, but this has a 10-segment genome. This has an 11-segment genome. But the blue cloud here is all 12-segment genomes that have arthropod hosts. So I'll just go through the phytoreoviruses are plant pathogens that are vectored by insects. The sidornoviruses, Southeast Asia dodeca RNA viruses, are human pathogens that cause encephalitic diseases, and those are vectored by biting insects. The cardoreoviruses are the Carcinus dodeca reoviruses. This is the recognized genus for most crab reoviruses, and they infect mitten crab, Ariaschere, Carcinus, the green crab, and Laocarcinus, the swimming crab. And then there's this mud crab reovirus here that infects Scilla serrata. It's an aquaculture species in China. And those authors propose that their virus doesn't belong with the cardoreovirus, but it defines a new genus called crab reovirus. So we were interested when we got our genome sequenced, where does our beast fall? Which of these genera does it fall into? And we analyzed the RNA-dependent RNA polymerase gene of CSRV1 and compared it to the available data for the other genera. And we also looked at the RNA banding pattern, which is the best we can do in terms of genome organization, compared that to all the other viruses in this group. And what we came up with is a tree that looks like this. This is based on the nucleotide sequence that CSRV1 definitely lands in the crab reovirus world with MCRV. But in our analysis, we also realized that one of the other mitten crab viruses belongs there, and that based on RNA banding pattern, not nucleotide sequence, but banding pattern, that really the taxonomy needs to be drawn like this. And that is that MCRV is over here in crab reovirus, here's CSRV1, but these other viruses that have been in the cardoreoviruses actually belong in the crab reovirus. So we've kind of shifted the weight now to this new genus actually now has most of the crab reoviruses in it. The founding member of the cardoreovirus, the mitten crab virus, ESRV 905, is still there. And remember that CSRV2 that I mentioned that we found, its banding pattern is just like the ESRV 905. So I think in blue crab now we've really identified one of each. We've got one in each of these genera. So I think that what we need to do now, the next steps are to get voucher specimens, and then changing the taxonomy of viruses is not a trivial matter. You have to go to a world body and kind of put in a proposal, and they argue it, and then people come up with evidence. But I think this is where we're headed. I think we're headed to this kind of taxonomy structure. Okay, so finally I want to get into what else the sequencing the genome tells us, and it has to do with sequencing the genome of viruses from different parts of the world. I talked so far, I've just talked about crabs, or virus from crabs in the United States. But thanks to a collaboration with Ana Paula Vinagre at University of Rio Grande do Sul in Brazil, we got crabs from down there from 2013, and I just got more crabs from there in 2015. And CSRV1 is present there. It causes lethargy and mortality, and we see it in both of their samples. It's at 30% prevalence. This is about 30 degrees south of the equator, so it's a similar habitat to what you might find on the Carolinas. In fact, if you stand in a salt marsh here, you could convince yourself you're standing in a salt marsh in the Carolinas. Very similar. But what can we do with this? We did sequencing from viruses isolated from each of these locations. We sequenced part of the polymerase gene, and we compared them to each other. And one of our questions was, when we found the virus down here, was, is this like one of these fast-moving global pathogens like you see in aquaculture species where you find it in one place and suddenly everyone realizes it's already everywhere? And it is. We think it is everywhere, but I don't think it came there recently, and here's why. This is based on sequencing of the RDRP gene. The U.S. Atlantic coast, we can cluster strains of the virus from Massachusetts and New York into one little arm of this phylogram. And then we have Virginia, Maryland, and Florida kind of clustered together on another arm of the phylogram. And then there's a pretty long branch over here to Brazil. So this suggests to me that the last common ancestor of the Brazilian strains and the American strains was quite a while ago, and it's unlikely that it came over with a person or crabs being moved from one place to another. So I think it indicates that what we have is a virus that's part of the natural history of the crab throughout both of the Americas. And now this gives you the opportunity to look at the virus as a co-evolved genetic marker that you can use in conjunction with the crab genetic markers to understand how crabs got where they are and when they got where they are, and what the barriers are between the two populations, between those populations. And I'll try to get into this. For example, if we think about the life history of the crab, larvae travel in the coastal zone, but they're sensitive to, they can't move fast. Right? They really at the whim of currents. And they're sensitive to low salinity. So there are certain places larvae can't get to just because of that part of their life history. And that is, if you think about the Amazon plume here, the Amazon plume carries fresh water, and it becomes low salinity water, along the surface, and it glides out. I mean, some of the tracer studies of the Amazon plume show floats, tracers that make it more than halfway to Africa. So that the plume has a really huge influence. But coastally, where you have crabs, if you have crabs along here trying to breed, a crab that's here and releases larvae, those larvae are never going to get down here. So crab in Guyana is never going to make it to Fortaleza. At least the larvae isn't, because it's going to hit the Amazon plume, which is going to advect it out into the Atlantic Ocean, or it may die because it's a low salinity lens of low salinity water on the top. On the other hand, a crab, a juvenile or adult crab, could simply decide to walk or swim across that. They're strong enough swimmers, they can swim across the current, or they could walk on the bottom. So if you think about the genetic consequences of that the genotype of the larvae aren't going to go from here to here because they can't get there. But the genotype of a virus that's carried by an infected crab could get from one side to the other simply by walking with the crab that can do that. And then the converse of that would be if we look at a place where larvae can travel and a swimming or walking blue crab can't. And this is the instance here. If you look at Florida and Cuba, there's a very deep straits of Florida between the two. And so crabs are benthic organisms. It's jury's somewhat out, but they're not known for making long-distance migrations as swimmers on the surface of the water. They tend to make long-distance migrations on the bottom. But it's too deep between Cuba and Florida for an adult blue crab to do that. But if the currents are right, you could have a larvae do that quite easily. I mean, it's less than 100 miles. So in that case, you could have the host genotype, the genotype of the larvae, are going to go from one place to another, but an infected crab won't. And so if you think about sequencing the genetics of the virus versus the genetics of the crab in different locations, you're going to get different measures of how they got there. So it's going to give you different information about how things move in the ocean. So I've worked with Lewis Plow at the University of Maryland who's a crab geneticist. He's a geneticist, and he's actually done some crab genetics, taking crabs from Florida and Massachusetts and showing that their genotypes can be kind of separated, but they overlap a bit. And then he looked at these Brazilian crabs and saw that they're really out there. Okay? And we're now working with physical oceanographers who are taking the latest Lagrangian physical oceanography models and trying to pair that with our genetic data. So this is sort of a nascent collaboration where we have the stars indicate locations where we have people that are willing to collect crabs for us. And we're going to be taking crab genetic data and sequencing the entire genome of the virus from crabs in all those populations and comparing those and trying to connect it with oceanographic currents as we understand it to come up with a really comprehensive model of connectivity. And this is just this November, I visited Brazil and met with our collaborator there and got a new collaboration started with somebody in Aracaju who's an expert on crustacean taxonomy there. So I'm hopeful that we can do this, but this is one of those aspirational slides. So if anybody's reviewing NSF grants out there, watch for this. So I think that's the end, and I just want to get back to the main point I wanted to make, which is this virus has the potential to affect blue crab abundance. This virus and other pathogens along with it. And just to remind you, I've talked a little bit about how this has implications for fisheries management, virus taxonomy, and population connectivity. And I've talked about it as me some of the time, but it's really, I'll finish this. Where we're going with this. So I talked about the fishery interaction, the collaboration with the South America and Caribbean. I also have two contexts in the Mediterranean. So the blue crab virus may or may not be in the Caribbean already, and that's a story that needs to unfold. But there are a lot of blue crabs in the Caribbean. And so I have collaborators in Israel and Italy providing crabs from there to try to understand if we have the virus there. And if anyone's interested, we found CSRV1 can infect other crustaceans in the food web. We've not seen it in the wild, but we know that those animals can be infected in experimental studies. So the food web is another place we need to look. Okay. I have to thank my graduate students. Emily did a huge amount of the work that I talked about. Matt Spitznagel is about to do more work than he knows on the fishery stuff. Behind him there, well, actually, here's a picture of Faith. She's actually my high school intern that's been with me all year, and she's figuring out the mutation rate in this virus to try to understand, get a molecular clock for those studies that I talked about at the end. And over each summer, we have really great, energetic interns who come in and work on short-term projects for an intense period of time. So I have to thank them. I'm not going to go through all the people I need to thank at other centers, but I will mention the funders. Almost all of this work was funded by different arms of NOAA, whether it's National Marine Fisheries or Ocean Service or the EPP. And with that, I will take questions if there are any. Thank you.
So I'm curious, when you're talking about prevalence, I'm assuming you're talking about detection of the virus itself. How does that translate to pathology? And do you have an indication of the pathology that's caused by the virus in the crab? Because what I'm getting at is more like, are there certain crabs that are carriers and don't really get sick versus ones that succumb to mortality?
I don't have specific information on pathology. And a lot of it is just the limitations of what I'm an expert at, but also a reluctance to spend the effort, because what I've found in these comparisons with Gretchen Messick at the NOAA lab is that we can look at animals, the same pieces of animals in our two ways, her histologically and me at the molecular level, and we see very different things. She sent me sets of animals where I'll pick up 10 to the 8th copies of virus per milligram of muscle tissue, and she'll look at that tissue on histology, and it's completely normal. She'll know ahead of time that that's the viral load I see. And so, but then if you look at the electron microscope level, you'll see a lot of viruses. It's just that somehow the virus doesn't create a lot of abnormalities at like the H&E staining point. I don't know if that's what you meant.
Trying to understand what kind of pathology is being caused by the virus.
Like behavioral and what systems are breaking down, or?
Well, also in terms of, you talk about implications of population management. Are all crabs being affected the same way? Finding a virus doesn't necessarily mean disease, is, in my mind.
Quite right, and that's why it's so important for us to get that tag and recapture study funded and completed, because that's my best attempt at seeing if there's a relationship between the virus and an impact at the population level. Does that make sense?
Yeah.
That
And there are possibilities too in genetic response to the threat. Are there areas where they're resistant or
yeah. On that note, I think this virus mutates so fast, and in fact, when you infect a crab with the preparation of the virus, so you're really infecting it with a cloud of genotypes. So I think the virus evolution is so fast that who can know? You may get resistance in the crab, but the virus is very adaptable. Yeah.
You said you didn't see any correlation between the virus and environmental conditions, but did you look if there is any correlation between the presence of the virus and the presence of other parasites or pathogens for the crab?
We have not, and that's a fantastic question. So the question was whether there are other pathogens or parasites that co-vary with the virus. And I didn't explain to you that the pathology of the virus is that it infects the hemocytes, that one of the primary targets of the virus are hemocyte cells. Those are the immune cells. And so we see that animals in end-stage infections become septic. They have a lot of bacteria in their blood. So I can imagine that if they're opening up a susceptibility to other things, so that may be an interesting place to go. Yes?
Are there any strategies to modify the water handling systems and the shedding tanks to minimize mortality?
Yeah, to minimize mortality. Yes, yes. So what we found is that in experimental infections, crabs die very quickly at higher temperatures. And I think that keeping your temperatures below about 25, 26 degrees would be a great way to improve survivorship. I'm thinking of things that at a practical level, these artisanal crab shedders could actually do. And I think keeping their water cooler would be one. That's probably the biggest.
And recirculating, perhaps with UV treatment or something like that?
Yeah, I think that the rate at which crabs contract and die from the virus is a lot slower than the shedding, the time that they spend in a shedding system. So I don't think they're contracting it there, but I think that if they have it, it's exacerbating it. You're right. Recirculating systems would solve two problems. You can probably do a better job at temperature control if you do that, and then you don't have the effluent that may be going into the environment.
So presumably, if humans or others eat crabs that have been cooked, there's no problem. But what about sushi?
I don't know. I don't know. So one, I think that reoviruses tend to be relatively host-specific, but I think I gave you 3 examples of reoviruses that can live in an arthropod and a mammal. I've certainly.
Why not?
Yeah. But if you were to eat it raw, I suspect you'd have no problem. I've certainly worked with enough of the virus that I'm either seropositive or, Yes.
Is the virus affecting other invertebrates such as shellfish in the bay?
I don't know. I don't know. What we know is that we can inject it into other crustacean species and see, in some cases, we can see it replicate in other crustaceans. I haven't searched in anything other than crustaceans in the food web for the virus, and I haven't seen it in anything else.
I was just asking, since the cow-nose race is been demonstrated is not the case for the base column decrease, I was wondering if this virus could be the case.
I don't know. It wouldn't surprise me if it were a virus. Yeah. Sorry. Yeah.
Okay. I was wondering if you had any ideas for what affects the natural variability of the outbreaks. That you were saying how there's differences between sites along the coast, and there was differences across years. And I'm wondering if you have any ideas about why that is.
I don't have any specific ideas. I guess I would just sort of go back to, the common wisdom is that you can have density-dependent phenomena, so that if you have times or places where crabs either just by happen chance you have a high density and a lot of interactions, you may get a lot of disease, or maybe sort of systematically, like seasonally dependent things where you often get crabs all in one place, you get disease transmission. Combine that with stresses that we don't understand, probably temperature stress among them, you may get a higher detectable level of virus. That's about the best I can do. Yes?
How long do viruses like this retain viability in the water or in the sediments?
I don't know with this one. Reoviruses have been shown to be viable for hours to days in water. I don't know sediments. In the water, viruses can be inactivated by lots of mechanisms. Sunlight is a really great inactivator, just ultraviolet light. I haven't delved into trying to count viruses in the water, but I now have some methods. Actually, someone put out a very nice paper on efficient and cheap methods for concentrating viruses from the water, and I think we're ready to look at that. Someone in the back was.
I don't have any intuition in terms of virology. The concept of infectious dose suggests a tremendous amount of nonlinearity in exposure to rapid replication. If something gets if you get a dose in the environment, a crab gets a dose in the environment, do they if it's below an infectious dose, do they carry it, or do immune responses take it down? Is it sort of like an on-off switch in a sense? If you see it, is there a risk? You know what I'm getting? Maybe you can ask a better question than
right.
that.
So no, these are. So this is going to be what, like the 9th time I said I don't know. The literature on infectious dose in shrimp, say where people have really studied viruses, is that there is an infectious dose. It's different for every virus, and that I think there's recent literature that's showing that shrimp actually do have an immune response, and it can be relatively specific. So that I think the implication is that they'll be challenged with the low dose of a virus, and I don't know if they've addressed whether they carry it, but then a second challenge with a higher dose results in no pathogenicity. So there's sort of a priming effect. I didn't quite answer your question.
Well, I guess the question is
are there carriers?
If you're measuring it, is that a significant risk if it's way below what would represent an infectious dose?
So in our aquaculture study, if we could detect the virus at all and held them in captivity, that crab will die in 2 or 3 months, in under 2 or 3 months. In crabs where we could never detect the virus, they would live 6 months. So I think, at least in our aquaculture setting, finding the virus at all shortens their life. But in the wild, we don't know. I think
Does that suggest to you, because there's exposure to the whole population, does that suggest that they're able to get rid of the virus
I think it could.
if it's below an infectious dose?
I wouldn't be surprised. Yeah.
You showed us a phylogenetic analysis of the RNA dependent or natural arrays of the virus, but you have the complete genome of many of these. So do you have identified genes or regions of the genome that are under more selection pressure than others?
Yeah. So the question is, if you compare a whole genome of this virus to a whole genome of a related virus, do you see parts of the genome that are evolving faster? So definitely yes, the RNA-dependent RNA polymerase is more conserved. The segment that we fortuitously picked as our diagnostic segment is one of the faster evolving if you compare it to related viruses. So yes.
Start of the capsid, or?
We don't know. Of that genome, we can only identify 3 proteins that we can identify a function for.
One last question. And
Yeah.
then
So going back to that point on the temporal variability of infection, do you think that short-term stressors like a hypoxic event could affect the infection rate on blue crabs, at least in the bay?
We've tried to look at hypoxia with the success of an experimental infection or the rate at which they die, and we didn't see a change. But you never know. Crabs are really resistant to hypoxia. I mean, the animals I have in the Aquaculture Center now came up with me in coolers from Florida. They were in a cooler for 9 hours. They're fine. So I don't know. I'm guessing hypoxia is not going to be a biggie. I think if you look at environmental variables, I'm going to look at temperature first. So yes, environment, probably not hypoxia.
Okay, thank you, Eric.
Thank you.